Hello, IB psychology students. This is my second video on the biological perspective. And my name is Linnea Karlberg. Today we're talking about localization of function. If you pause the video right now, you can find out what the IB requires you to know. I think it's interesting to look back in history because this theory kind of originated from phrenology. Phrenology refers to the fact that measurements of the brain was believed to predict our character traits. You can look at this picture right here. I find it very interesting. But of course today, it is quite different. Localization of function refers to the theory that a given function is localized in specific areas of the brain. So simply that certain brain structures, or brain regions, are the genesis of particular behavior, cognitions, and emotions. And people have different thoughts and beliefs regarding this theory. Looking to some strengths or some support to this theory, we can see that brain scan evidence provides support for localization of neurological function. However, there are a lot of weaknesses to the theory. First of all, it's very biologically reductionist, meaning that they're attempting to take a complex human behavior and narrow it down to one structure. It just doesn't seem quite likely that easy. We can look at Lashley's exponentiality theory, which hypothesizes that basic motor and sensory functions are localized, but that the higher mental functions are not. We can also see that functions are not localized to just one region because other regions can take over a specific function following brain injury. And some argue that it should be more important to investigate how the brain areas communicate with each other, rather than on simply focusing on specific brain regions. Another thing is that it fails to take into account individual differences. For instance, Heresy found that women have proportionally larger Broca's and Wernicke's area than men. So these two are brain areas. Okay, the brain area we're going to focus on firstly is hippocampus. So this is a small curved formation in the brain, which consists of gray matter covered in ventricular surface with white matter. And it's associated with the limbic system as well as formation of memories. You may remember this study, Maguire et al. It's very common to talk about it in psychology. And it talks about the taxi drivers develop more pronounced hippocampi as a result of navigation of complex streets. It also talks about neuroplasticity, which we'll go into in a later video. This was a natural experiment, and it was implemented through MRI scans on 16 healthy right-handed male London taxi drivers and 50 healthy right-handed gender-matched controls who did not drive taxis. They also recorded the amount of years driving taxis. So what they did was they measured the size of the hippocampus of both of these different people. And, as I mentioned earlier, hippocampus has been found to have an importance in memory. So it was found that spatial maps, or memory, alters brain with expertise, because the anterior part of the hippocampus was bigger in the controls, and the posterior part of the hippocampus was bigger in the taxi drivers. So hippocampal volume of the right hippocampus in each taxi driver correlated with the amount of time spent as a taxi driver, positively in the right posterior and negatively in the right anterior hippocampus. So a larger posterior hippocampi suggested a more proficient at spatial memory and navigation. It also showed that environmental demands of being able to navigate a complex structure of streets allowed the drivers to develop more pronounced posterior hippocampi than the control subjects. Overall, the study's high in ecological validity as it looks into an actual real-life situation. Also, a control group was present used for comparison. The variables were clear and identified, and there's been triangulation of this theory. They've tried it as well with bus drivers and found similar results. However, there may be some confounding variables at play, especially when it's so related to real life, as well as the sample is quite small. And as there was a manipulation of the independent variable, they cannot identify a cause and effect relationship. Another theory we can look into, a study, is by Scoville and Milner and Corking et al., who looked into HM's memory loss. So this was a cognitive longitudinal study on an epileptic patient. So what they did was interviews, cognitive testing, observation, and fMRI. Following that, this patient had removed medial temporal lobes and part of the hippocampus during surgery. The seizures went away, but HM suffered from partial retrograde amnesia, meaning he had little memories from 11 years prior to surgery, as well as anterograde amnesia, meaning unable to detect that time has passed, but he did know a few facts, like who Elvis Presley was. First of all, the fMRI allowed to effectively demonstrate the long-term memory formation being localized in the hippocampus and hippocampal gyrus. So it wasn't only the hippocampus. And so they stated that hippocampus is likely involved in the formation of new long-term explicit memories, and that refers to like facts. Since this was a case study, it looked into quite an extreme condition, and that allows for great insight, which provided us with information on localization of certain elements of memory creation. This study also had methodological triangulation and research triangulation, which increases the validity of the results. Another triangulation is another study known as clev-wearing, which many people have talked about in their psychology class. And there was also anonymity. We didn't know who HM was until he died. However, one can question the consent. Can you really get permission from someone who doesn't remember? And there are some issues with generalizability when it is a case study we're talking about. The second area of the brain is called the amygdala, and it's a roughly almond-shaped mass of gray matter. And it's localized inside each cerebral hemisphere. The amygdala is associated with emotion, and specifically fear, like also cringing, freezing, tremors, and memory. And a study of this area is by Feinstein et al. This was a case study on a patient called SM who perceived lack of fear. So SM had a rare genetic disease. It's called Urbach-Wiete disease. I'm sorry if I pronounced that wrong. So this person had lesions in temporal lobes. And this led to significant damage to both her amygdalas. So what they did was they exposed SM to live snakes, and they knew before that this person had a hatred of snake. But she would still hold them, driven by the curiosity. They also toured SM through a haunted house, made her watch fear-inducing films, and went through a 3-month-long interview. And they found that this person didn't seem to be able to experience biological fear. And fear, it's an evolutionary benefit. It provides safety and fight or flight. So SM would end up in a risky situation, often victim of muggings and violence. But it allowed them to suggest that the amygdala is a critical brain region for triggering a state of fear when an individual encounters threatening stimuli in an external environment. This study allows for clear illustration of localization of function, as this person could experience all emotions in the same way physiologically healthy individuals do, just not fear. There was also consent obtained from SM, and as before, we don't know who SM is, as anonymity. However, Dr. Leduc suggests that this was quite an oversimplified view on the biological ethology of fear. He suggests we should more look at the amygdala as a contributor to human defense survival circuitry. And that emotion, it's quite a large cognitive component. So maybe this person just simply reasons that snakes aren't life-threatening. If there was a car coming, it's likely she would move away. There's also little evidence supporting this. It's more makes sense that the amygdala regenerates the conscious experience of fear. Another thing with fMRIs is it's quite expensive and time-consuming, and there's issue with generalizability as it is a case study. And one could also question the protection of SM from harm, as they made her do all these crazy things. There's another study that looks into both the hippocampus and the amygdala in fear conditioning with rats. So this is a lab experiment. There was a control condition, which was non-lesioned. There was one condition where they had the amygdala lesioned and one where the hippocampus was lesioned. So they attempted to condition the rats to associate the sound of a tone with an electric shock. So the rats whose amygdala was lesioned, they had resistance to fear conditioning, and rats who had their hippocampus lesioned could be conditioned to fear the context in which the shock was administered, but not the cue of the shock. So this made the researcher believe that the role the amygdala serves in fear conditioning is associative, and the hippocampus plays a role in one of the relying sensory information. And emotional memories are not from one area of the brain. The hippocampus and the amygdala work simultaneously together to create them. This study showed significant implications to help understand memory and emotion, and it couldn't have been acquired if non-human animals were not used. There are also high control of variables and an easy replication. However, once again, it's quite a reductionist view of fear, and the ecological validity is high, since not even humans are used. Can we really apply this to humans then? That's definitely something to question, and if you're an HL student, you will look into more animal studies. Thank you so much for watching this. If you have any questions, feel free to comment. See you in my next video.